Good day to each and everyone. Today I will be presenting our research entitled Biological and Environmental Profiling of three Philippine Tetrastigma species in Northeastern Cagayan, Luzon, Philippines. Tetrastigma is a member of the Vitaceae family, which has approximately 95 species worldwide, and it was distributed throughout the Southeast Asia, Australia, India, China, and others. And it was locally known as Ariwat in Ilocano, Alupidan in Bisaya, and Ayo in Tagalog. It was also reported to grow in various habitats, such as in the low to high elevations, hillsides, valleys of shady, moist primary rainforest. And the plant has a woody vine. It has a climbing structure, and it has brown ovoid sour fruits. And the relationship of Tetrastigma species to other plants is that it is a well-known host plant of Rafflesia. In terms of the species health and medicinal properties, Tetrastigma species were reported to contain various phytochemicals and pharmacological properties. It was shown in this figure that Tetrastigma glabratum and leucocephalum contains various phytochemicals such as alkaloids, phytosteroids, saponins, terpenoids, flavonoids, and tannins, carbohydrates, and others. And for the Tetrastigma hemsleyanum, it was reported that it contains various pharmacological properties such as anti-proliferative, anti-tumor, antivirus, anti-inflammatory, anti-diabetic, and others. However, in terms of the pharmacological properties of Philippine Tetrastigma harmandi, it was reported that it has anti-scabies and diuretic properties, and also reported to treat urinary diseases. Our study wants to add baseline information on the distribution, habitats, and growing environment of three Philippine Tetrastigma species, namely Tetrastigma ellipticum, harmandi, and loheri in Northeastern Cagayan. Also, we want to add additional morphology characters, specifically for the Tetrastigma loheri, where we compared the type of leaves of the species after asexual propagation. And also, we've profiled the phytochemical content and the antibacterial potential of the three Philippine Tetrastigma species. In terms of the nutritional content, we want to add information on the nutritional content of the two common edible species, the Tetrastigma harmandi and the Tetrastigma loheri. To contribute to the database about Tetrastigma species in the Philippines, the study determined the following: existence and distribution of Tetrastigma species in various municipalities of Northeastern Cagayan, and it hopes to look into the Tetrastigma species growing in the protected areas of Northeastern Cagayan, documentation of the morphology and growing environment to include the soil characteristics and plants that grow within the Tetrastigma environment, investigate the phytochemical and antibacterial properties of the three Philippine Tetrastigma species, and also determine the nutritional content of the two common edible species, your Tetrastigma harmandi and Tetrastigma loheri. Our methodology actually goes like this. We characterize the Ayu species through visiting various field sites in Northeastern Cagayan and identify the species morphological characterization. We conducted also environmental analysis, specifically on the soil properties of the three Philippine Tetrastigma species, and at the same time, where the habitats of these three species, and at the same time, the various associated plants that grow within the Tetrastigma species environment. Also, we've conducted phytochemical screening, microbial sensitivity tests, and nutritional and mineral analysis for the Tetrastigma species. For the qualitative phytochemical screening, we tested the presence of alkaloids, flavonoids, phenols, terpenoids, anthocyanin, tannins, steroids, and saponins in the three Tetrastigma species. In terms of the microbial sensitivity test, we've tested the antibacterial properties of the species against Staphylococcus aureus, E. coli, P. aeruginosa, E. fascium, K. pneumoniae, K. aerogenes, and A. baumannii using paper disc diffusion method. For the nutritional and mineral analysis, we've tested the crude contents, the macro and micronutrient contents of the Philippine Tetrastigma species. In terms of the results on the species morphology, I want to emphasize here that the Tetrastigma loheri species has actually three variations in terms of the young leaf structure, where the species that were found in Mount Kagwa has actually green and reddish young leaves, whereas those that were found in Palawi Island protected area has actually reddish yellow young leaves. Okay. In terms of the geographical distribution of the species, the Tetrastigma harmandi were evenly distributed throughout the throughout the northeastern Cagayan map, whereas the T. ellipticum and loheri species were only found in Mount Kagwa, whereas other species of T. loheri were found in Palawi Island and Buakag Mountains of Sierra Madre. In terms of the habitats and growing environment, it was shown here in this table that Tetrastigma ellipticum and harmandi were found in various habitats, such as in high elevations, in lowland elevations. For the Tetrastigma harmandi, it was found in the residential areas, near coastal areas, nipa plantations, agricultural lands, island shorelines, secondary growth forests, and others. For the Tetrastigma ellipticum, it was found only in Mount Kagwa. Okay, in terms of the other plants that growing within the Tetrastigma environment, it was shown here in this table that the species were actually growing together with some native and endemic tree species. Okay. In terms of the loheri species, it was found only in Mount Kagwa, Buakag Mountains, and in Palawi Islands. And also, they were found to grow together with various native and endemic tree species. But for the Tetrastigma harmandi, most of the species were found climbing together with ipil-ipil and kakawati trees. In terms of the soil characteristics, I just want to emphasize here that Tetrastigma ellipticum and Tetrastigma loheri, which were found in the forest, were growing in the soils with highly acidic pH and a high organic matter content. For the Tetrastigma harmandi species, it was growing in soils with higher pH, soil pH, and lower organic matter content. In terms of the trace elements and macronutrients, it was shown here that the species thrive in various levels of nutrients in the soil. For the phytochemical and antibacterial properties, it was shown that among the three species, the Tetrastigma ellipticum contains the most number of plant secondary metabolites tested present, which are the following: alkaloids, flavonoids, phenols, terpenoids, anthocyanin, tannins. But the only species that contain saponins are actually your Tetrastigma harmandi. Steroids were not detected in the three Philippine Tetrastigma species. In terms of the antibacterial properties, only the Tetrastigma harmandi has antibacterial potential, against Staphylococcus aureus, and a slight inhibiting activity against Klebsiella aerogenes. The two mountain species, your Ellipticum and Loheri, do not have antibacterial potentials against the bacterial strains tested. In terms of the nutritional content of the two edible species, it was shown here that the Loheri species contain more food contents as compared to your Tetrastigma harmandi. However, in terms of the macro and micronutrients, your Tetrastigma harmandi contains more nitrogen, phosphorus, potassium, calcium, uh, manganese, iron, zinc. However, in terms of the copper content, Tetrastigma loheri contains higher amount. Our results or our research highlighted that the Tetrastigma loheri species encountered in Mount Cagua had red to light green newly emerged leaves, and encountered in Wacag Mountains had light green newly emerged leaves, while those encountered in Palawi Island had red to reddish yellow newly emerged leaves. Tetrastigma harmandi growing in various habitats were widely distributed throughout the northeastern Cagayan, while Ellipticum and Loheri species were only found in the mountain forest of northernmost part of northeastern Cagayan. Okay, most of the plant species growing within the Tetrastigma environment were native and endemic trees, and the mountain species of Tetrastigma grew in extremely acidic soils with high organic matter content. More secondary metabolites were detected in Tetrastigma ellipticum, and Tetrastigma harmandi was the only species to have antibacterial properties. Between the two edible species, Tetrastigma harmandi contains more macro and micronutrients, while Tetrastigma loheri contains higher crude protein, higher crude fat, and higher crude fiber contents. For research opportunities, the potential of utilizing the species in product development for medicinal, therapeutic, and industrial purposes are opportunities. The present findings on the habitat conditions of the Tetrastigma species will guide the future researchers in investigating their growing environments in other places or localities or ecosystems. Moreover, the concentration in various plant parts and the specific identities of the phytochemicals present in the three Tetrastigma species should be further explored. Initial findings on the antibacterial potential of Tetrastigma harmandi against Staphylococcus aureus will guide future researchers in evaluating the efficacy of medicinal Vitaceae plant extracts in treating various diseases posed by the pathogenic bacteria. I would like to emphasize my sincere gratitude to the following agencies, people, and institutions that have contributed a lot to the success of this study. So that ends my presentation. Thank you for listening.